So I work in antibacterial lead discovery, as she said, at Novartis. And so the way that I got started on this was really my fascination with these single-celled microbes. To me, it is amazing just the diverse ecological niches that they can occupy, places that no other living organism can survive. Yet they found a way to live there, produce, and thrive, as well as their role in symbiosis with many other eukaryotic species, and then how we've been able to kind of learn how to use them for our own benefit in many applied functionalities. And the reason I got started on microbes was really due to my high school biology teacher. So like many of you, he was my inspiration. He wrote NSF grants that he took a back stock room and he was able to get a small incubator in there and an autoclave that we could use to sterilize our media. And my first project that I worked on for him was photoreactivation of Serratia marcescens, this little red pigmented benign bacteria. And that really started me on my path. I love working in those little simple bacterial genetic projects, and for the remaining years of high school, that's what I did. I competed in international science fairs, and I had him as my mentor. He was the person who taught us how to question things, how to ask why, and really be amazed at the answers and the whole process that went into understanding these organisms. Through some of his connections, I ended up at North Dakota State University, so I am from the Midwest, the big flyover area that most people are familiar on the East and West Coast. But at North Dakota State University, I continued in my passion for microbiology. I explored lots of other sciences, but microbiology was always the thing that resonated with me. It was always the thing I understood the best out of everything that I studied. And from there, I went to Iowa State University, yes, another popular place in the Midwest, where I worked on staphylococcal genetics under Dr. Peter Patty. And Staphylococcus at the time was thought to be kind of a second-rate organism. It's a gram-positive that, it's interesting, but everybody was studying E. coli. E. coli was the organism that everybody knew. It was, if you could learn about an E. coli, who cared what happened in other bacteria? E. coli was all that was important. But it turned out when MRSA came out, then everybody cared about Staphylococcus aureus, and our lab became the lab for bacterial genetics. But I also wanted to know what goes on in the cell besides the genetics. There's other factors that control, and how do they respond to all those different environments? And so my postdoc was at UC Davis, where I studied signal transduction in bacteria. So when they sense their environment, how does that change? They can respond to so many different stimulus, yet they're a single cell. No nucleus, just chromatin and a bag of enzymes. And so that was the whole process. And through this foundation that I had in microbiology, I ended up with my first job in biotech here in the South Bay, which was at Microside Pharmaceuticals. And in a biotech, you start with one job, and it ends up becoming many different jobs. You learn to wear many hats. But in a biotech, what we were taking was my foundation in staphylococcal genetics, and we were trying to look for antibiotics for MRSA. So the theory was, well, these resistant organisms need to have something to put them into check. And at that time, it was the huge genomic era, and if we could just sequence all the genes, we would understand how they work. We would find all the drugs. Bing, bang, boom, there you go. It wasn't that easy. So when I started with my staphylococcal genetics, I soon moved into being able to screen for those new antibiotics. And that's where the pharmaceutical industry hooked me. I loved that antibiotic treasure hunt. I loved learning about all the different technologies and processes that it took in order to be able to really figure out how are you going to be able to modulate and control those bacteria so they can't cause infections. How do you do that? And so it led to a whole series of 2 developments. This small biotech company had a collaboration with Pfizer, and I was one of the main scientists on that collaboration, and when it was over, they recruited me, and so I went to Big Pharma, which is a very different research environment from biotech. And at Big Pharma, you can get really niched, and you can go deep, and that is a plus. But I never seem to be able to go deep on everything. I always end up having to be spread across all new different exploratory areas. And so a lot of them that we went into was trying to better understand how compounds were interacting with the targets in the cells. And so it led to a lot of structure biology work, which was a completely different science than what I was trained in. And I still probably am not an expert in it. And when you work for a big pharma, you have to be prepared for a lot of mergers and acquisitions and changes, and you learn learning agility, change agility, all kinds of agility things. And so while I was there, I did a brief stint, I call it my neuroscience sabbatical, where because I had been so involved in exploratory activities, they had me also work in some of the neuroscience exploratory activities, which was also fascinating. I love learning how the brain functions, how all these different diseases had so many different complications, and how the human population, just like the bacterial populations, were diverse in a different way. And it brought a lot of analogous thinking, a lot of orthogonal thought for me, but my heart was still back in antibacterials, and Pfizer unfortunately decided to abandon antibacterial research. And so when I was offered the chance to come home, I took it. And so I joined Novartis here. We're located in Emeryville, and now I'm back in antibacterial discovery. So at Novartis, there's still an opportunity for me, if I wanted to explore other disease areas, but I'm still really focused on antibacterials. And the one thing I like about Novartis, if I had to compare the 3 different pharmaceutical companies that I've worked through, I would call Novartis Mama Bear. Biotech's like Baby Bear, Pfizer was definitely Papa Bear, and Novartis is Mama Bear. And the reason I say it's Mama Bear is because it combines all the elements of what I think we go into this type of research. We really want to bring medicines to the patients who need them. I mean, everybody I work with, if you ask us, what do you want to get out of your career? It's like, I want to get a medicine on the market that's going to help a patient. That's why I'm here. That's what I want to do. We want to be creative. We want to do innovative things. We want to be able to explore and use the science around us. We don't want to feel like we're in an auto industry where we're just putting parts and making the same machine and hoping that it runs differently. We want to be creative. We want to explore. We want to learn new things. And so part of that is working with other scientists and having a more collaborative framework, which allows us to do that. And Novartis brings all those things to my work. Why do I like antibacterials? Why does it resonate? Not just my fascination with microbes, but I think antibacterials or antibiotics have really enabled a lot of the modern medicine that we have. When you think about all the more complex things that they can do in the hospitals and for therapeutic areas, a lot of them, the bacterial infection is the complication. The bacterial infection, you can go in the hospital and have a procedure, but you can get a nosocomial infection that's going to maybe circumvent that outcome. And so having the right antibiotics for those diseases so that you can, get back to your life, get back to the therapeutic outcome that you need it to is what we want to try to be able to enable. And the need that we have for new antibiotics really stems from the resistance that's out there. So bacteria, as I said, are very evolutionarily capable. They've survived for thousands of years and they can adapt to many different things. And one of the things they adapt really readily to is to the antibiotics that we give them. So it's kind of like a race against time. Can you keep your drug therapeutically active long enough for versus the bacterial evolution. And there's always new pathogens that are emerging, as well as, some of these procedures make the patients more immunocompromised. And so by lowering their defenses, they're just more vulnerable to those bacterial infections. And so that's one of the consequences of being able to have that procedure is now you're going to be a little bit less compromised or more compromised. So when they look at the statistics for why antibiotic resistance is such a problem, some people say, well, 23,000 deaths really isn't that bad. I mean, there's other things that are worse. But when you think about it, it's like a jumbo jet crash every week. So if you imagine a plane going down every week, that's kind of what it would look like. But the more alarming number, in addition to that, is just how many different illnesses are a result of those resistant antibiotics. And this number is probably not even very solid because it's always hard to really tease apart if somebody went in for a surgical procedure and got a bacterial complication and had a mortality. Was it because of the surgical procedure or was it because they succumbed to a bacterial infection in the process? So, but it's still, and it's an alarming number, and it's a big medical need. So, some of the challenges we face, as I mentioned already, that the pathogens can evolve very rapidly. There's a lot of natural selection that they have. And the bacteria itself, because it's not just the antibiotic, it's just the whole defense mechanisms they've evolved over time. Not just because it's an antibiotic, but it's all the other toxic things that are in their environment that these different defenses have evolved too. They have membranes that can help protect them. They have efflux pumps that just kind of pump your drug right out of there before it has a chance to do its job. And a lot of this is based on what's in their environment. When you think about natural products that they coexist with, well, a lot of the resistance mechanisms are because they've seen entities or they've seen things like that in their environment that they've co-evolved with. And sometimes we're selecting for those resistant species by removing the more susceptible ones from the environment. And that gives them that intrinsic resistance to just a broad range of different types of chemical molecules. The other thing is safety. If we were just trying to kill bacteria, we could just pour bleach on everything and it would be great. Bleach kills all. In fact, unfortunately, these bacteria are in your body, and you can't necessarily just pour bleach on everything that's in your lungs or your heart or your kidneys. So safety is a huge factor for us. We have to make molecules that are extremely safe. They have to kill the bacteria, but they can't damage the host. And there's a German philosopher that says the only difference between medicine and poison is the dose. So when you think about it, there's lots of things that you could take in small amounts that would be perfectly fine, but if you have to give a large dose, like what you may have if you have a really resistant bacterial infection, that can start to get to the borderline of now it's becoming more like a poison than it is a medicine. And so that's the fine balance that we're always trying to weave. And so that means that our compounds have to have some unique chemical properties in order to make them more safe than, say, bleach would be, which would be very simple to make and we could do all day. Okay, so the drug discovery process is really composed of two main sections. There's the lead discovery phase that I've been involved with most of my career, and then there's the clinical phases, which are more regulated by the FDA, and they revolve around the clinical trials for efficacy and safety. But I'm going to focus on this lead discovery phase because it's the part that I love the most. Okay, so we start off with, what do we want to work on? So I'm going to walk you through these phases, and it's going to be a little bit interactive as we go along. Don't worry, there's no right or wrong answers. There's no quiz. I don't have the answers. Everything's fair game. But in every place I've worked, the way that we decide what we want to work on is we interact with physicians. And at Novartis, our physicians that we work with on our teams are actually clinical practicing doctors. They have clinical hours, they're in the hospitals, they're seeing patients. The head of our disease area came from USCF, or UCSF. He's a practicing doctor. He really knows what patients go through. He sees them on a weekly basis. So he can give us a real world feel for what we're trying to do. And it really brings it home to us because he can relate to the challenges that he faces in treating patients. So what we ask of those doctors is, what are the diseases that are important? What are the gaps in the treatment? So what can't you treat? But where are you lacking, the appropriate therapeutic agent in order to treat that disease? And then what causes those diseases? So we have to understand that causative versus correlative association with the disease in itself. And at least for antibacterials, it's a lot more straightforward than it was for neuroscience. So I'm thankful for that. But then we have to break it down and we have to look at, well, if we understand what bacteria cause the disease, can we understand how those bacteria work, and can we figure out what to target? How do we modulate their growth, their activity, so that we can control them in a hospital setting? And that becomes a lot of cell biology, a lot of biochemistry, that we have to understand how things work in the cells. And that's a fascinating area of research for me. So once we have to form that hypothesis, what's our rationale for what target, what process we want to do to work on? From there, then we start getting into more nitty-gritty. So like if I were to pose to you, would you work on a target or a process in the cell that you knew really well, or would you work on a novel one? Any thoughts? Pros and cons for both? Just yell it out. Novel one.
Yeah.
I'm thinking because the process is more information, more data on the novel function than it would be the non-novel function.
Yeah, so if it's a known process, and by known I mean, I should explain it a little bit more, like there's a marketed drug for that. Obviously, there's something about that drug that doesn't work. Otherwise, we wouldn't be coming back to that target, that pathway. So we have to understand why it's not working, whereas a novel drug, we don't know anything about that. Whatever we discover could work. There's downsides to that too. Any thoughts about the downside for working on a novel target or process that you can think of?
It's hard to foresee any complications that could arise because there is no other product out there.
Yep, it's the devil you know versus the devil you don't. And there's many times where we'll stop a project because somebody will say, I don't know what's going to happen next. And that's a big risk. I don't know if we should keep working on this. There's so many unknowns. There's like obstacle after obstacle. You solve one question and then you got another. For how many years are you going to work on this? But you have to do what you can do. With bacteria, like I said, many infections are polymicrobial. It may not be just your pneumonia may not be caused just by one organism. It could be you have a whole bunch of stuff going on. And there are some infections that are caused by a single pathogen, like say CF, where Pseudomonas is more commonly associated with that in the later stages. So when you're thinking about that, in terms of the bacterial diversity, would you pick a process that existed in many different bacteria, or would you pick that single pathogen?
I'll go for the many.
Go for the many? Why?
Bigger market, potential market.
Yep, empiric use, the silver bullet. If you can give me a drug that wipes them all out, like bleach, you got yourself a deal. The commercial would be totally behind you. How easy is that to do, though? What's the downside of that?
Everyone's going for it.
Variation, variables.
Yeah.
Tested.
If it was that easy, it'd already be easy.
Yeah.
Yeah.
Well, maybe some could become resistant.
to that.
Sorry.
I was just gonna say, some kid, if you're using the same kind of disinfectant, then some kids become resistant to that and then survive past that. So, I mean, that's a possibility that can happen. Just like the antibiotics, you keep using the same amoxicillin or whatever, that if this doesn't get killed off, it can become genetically modified and become resistant to it, and cause other problems.
Yep. And your point?
Well, I mean, we do have penicillin and certain medications that work, really well. But of course, bacteria are known for, just evolving, right, running away. What is the standard?
Yeah, and the reason that like the first generation penicillins aren't really that effective anymore is they have evolved. And so you have first, second, third, fourth, now we're on our fifth generation penicillin-like drugs because they keep finding ways around it. And so, yeah, I mean, it's commonly conserved, process in the cell to have that cell wall replication, but they all rely on things a little bit differently. And to find a process that they are equally dependent on, that is so essential for all those different diverse species, that's hard to do because they are so different. And how they will survive in the host has different capabilities, so they have all these different tricks that they can play with masking themselves so they look like the host, but they're not the host, but they can evade the immune system. So it's really hard. It's like herding cats. Like, can you get everybody to just do what you want them to do? They don't do that.
It's our job.
Oh, go ahead.
So you mentioned that the penicillin were the fifth generation, and they're constantly evolving. Will they evolve to a place where you go back to the first generation and it'll work again? Does that make sense, my question?
No, because one of the ways that they've evolved, especially with the penicillins, is they don't really, well, there are target modifications for the actual protein that they bind, but they produce an enzyme that basically just destroys the compound. So they can produce this enzyme. If they see that compound, they can destabilize a ring, and now that can bind to the target anymore. And so if you're living next to somebody who can do that, well, they can do the job for you. You don't actually have to have that tool yourself. You just have to be buddies with some guy here. So it does get complicated when they're polymicrobial infections. And it's part of the reason why people think of things as cocktails. Well, if one drug can't do it, let's throw 2 or 3 at it, and that way we're covering everybody. And that gets complicated too. So another thing we have to consider when we're looking at the targets is the complexity of the function. So the bacteria are going to rely on different processes, and we kind of separate them into kind of 2 major groups. There's the mechanical ones, like that they physically do something. And then there's the enzymatic ones, which maybe they're taking a substrate and they're turning it into a product, and these are building blocks that then become higher ordered structures in the cell. So would you work on a target that was more mechanical, that required a complex in order to execute that function? Or would you work on that enzyme that just converted a substrate to a product all by itself?
What degree would you consider the research or the knowledge about how the different cells already interact? Like how much background knowledge or background information would you have to determine like how many different ways you can analyze what the function is?
I would say in the host, that's a growing area, because we're used to thinking of them as little organisms, so easy to grow on a petri dish, so easy to grow in a broth, that we think of just in their isolation, how we're going to work with them. And bringing that holistic view of, okay, now you have them maybe expressing different genes in the host than they are on your little petri dish where you're providing them a certain set of nutrients and you can kind of predict what their metabolism is going to be and the rate and the types of products that they're going to produce. It's a whole different game when they're in the host, and that's a really good point. That we'll kind of come back to a little bit later. But would you work on a complex or would you work on a single target?
Complex.
Why? Either one.
Complex, if plan A doesn't work out, you know a lot, you acquired a lot of knowledge to go to plan B. And also if it's somewhere down the road that drug doesn't work or it's time and you lose your patent, you get some other things in the quiver.
Yeah, that's a good point. How easy would it be to work on a complex? How would you understand what your drug is doing?
Hard.
By definition, it's not going to be as straightforward. You know, and a lot of it's kind of a paradox. A lot of the early discovery efforts were really on, especially after the genomic area, we got, okay, if I have a gene going to the central dogma, it's going to go to a protein, and I can understand the biochemistry of what this enzyme does, I can understand its kinetics, I can figure out how I can use these structures, I can get all this information, but the problem was that a lot of times these bacteria, since they're so capable of mutating and evolving, well, guess what? The first thing they figured out how to do is how to take your drug out of their active site, and so now you no longer can find a target. So a lot of programs we've had where we've had single gene targets have failed because bacteria, even before we could get to the market, figured out how to get around it.
Sorry, I was
Okay. So, and then, like I said a while back, you know, we have to be really concerned with safety. Even though prokaryotics and eukaryotics are very diverse species, there's still some commonalities that we have between each other, or enzyme families that have similar functions where they might look a lot like each other. So, and those tend to be the really essential processes because they are so core to their viability. So would you take a chance and work on something that maybe was shared with the eukaryote, or would you stick to only things that were prokaryotic?
I'm not a risk taker, so I probably would not have functional support.
Yeah. That's a safe bet. Sometimes we don't know what we really have in common with our eukaryotic counterparts. We may think we're really diverse because we're working on a novel pathway that only bacteria have, but lo and behold, the nature of the proteins, the way they're folded and the shapes that they have, our drugs now can recognize those mammalian counterparts that we think are very disparate in function. So we have to know a lot of mammalian biology as well as our prokaryotic biology in order to understand what those safety risks are going to be for the compounds that we're trying to generate and advance. So I've spent a lot of time in lead discovery on assays, and assays are really the platforms that we use to try to understand the biological functions. And in the early days when HGS first became, again, another savior of the pharmaceutical industry right after genomics, was once we have all these targets and we can make all these proteins, well then we're going to screen millions of compounds, and then we're just going to have drugs coming out of our ears left and right. And so the early assays were either like the natural products ones where we looked at whether you can just kill bacteria. So we would just measure bacterial viability. That's classic. It's the sure thing. Dead bugs don't mutate. But when we're looking at things from like that biochemical perspective, we were looking at things that we knew how to visualize, how to measure. And so they became colorimetric assays where we could measure the chemical reaction of one of those cofactors or the byproducts of that reaction and be able to see a difference. So it was always, can we see you know, what no effect is versus 100% effect, and then measure what's happening in between there so we can understand the activity of the compound and its potency. A lot of those assays really narrowed what we could work on because not everything could have a colorimetric reaction, or it could be coupled to something, but a lot of those systems that they had to be coupled to in order to generate that readout were things that then would confound the interpretation. We would find things that more affected the coupled reaction than the actual reaction that we were trying to measure. And so we've evolved. We're trying to get more sophisticated as well. And so some of the technologies we're using are like mass spectrometry, where we can actually go in and measure the substrate, measure the product, look at the consumption, look at the production, and actually see how things are being affected. We can go in and look at that biological pathway. We can measure the metabolites in the cell. We can see how modulating that process is really changing the function of that whole pathway. And we see, you know, a stop or an accumulation of intermediates along the way where we know we're inhibiting the process that we think we are. We also get into more cytometry type of things like flow cytometry, where we can look at populations of bacteria and not just at one single little thing. So we can see the whole range, the whole diversity, the heterogeneity of what's happening in the cells. Are they all responding to the treatment, or is there some small population that really is resilient and our compound's not doing anything to them? That gives us clues about how effective that might be in the future in terms of like what kind of resilient populations or resistant populations are going to form.
5 minutes.
Oh, okay.
Sorry.
And then there's like since I like pictures, morphologically we can see what does this actually do to the cell. So we can see, is it visually changing the morphology of that? So I touched on this a little bit. You know, the industry started off with natural products, which is still a fascinating area. There's biological warfare going out in the environment all the time. Can we understand how to use that, how to turn those into drugs, how to find the novel molecules that will be pivotal? We've spent a lot of time in chemical libraries, and that was the next phase after HDS, because we're just going to make millions of compounds, and in that diversity we're going to understand all our new drugs. Well, even that's limited. And now we're looking more at how do mammalians, since we're trying to fight, you know, mammalian infections, how do they defend themselves? Because not everybody's getting sick all the time. So we must have some defense. So how does our body defend itself? How can we understand what those entities do, and how do we employ them in a more industrial way? So I've talked a little bit about this, where our basic assay was to take compounds or whatever modalities, our biological system, and we look at different readouts. We need lots of robotics. When we were talking about screening millions of compounds, if you don't want everybody in your lab to get carpal tunnel, you're going to build a robot, and you're going to have a machine do the work for you. My lab loves those. So, and when you're screening millions of compounds, you get tons of data. And it's not just the data from your ask, which is a lot, but if you really want to make sense of the whole thing, it's how do you integrate all the different data from the different assays that you need to understand that compound's activity and the implications of it on many different facets of your system. So one of the things I'm not going to touch on a whole lot, but it has to do with the disposition of the compound in the mammalian body. So compounds undergo transformations all the time because your body's got built-in defenses of how to turn those chemical entities into things that aren't going to harm it, and it can excrete that. So that's a whole other element of the whole drug discovery process that we have to take into account as well. One of the tools that I really like, and I still like it, even though I'm more phenotypic these days than not, is I think structural biology really gives the chemist a good handle of what's going on. So in structural biology, they can take the proteins or the complexes that we're working with, and they can make these crystals and then get X-ray diffraction pattern from those crystals. And those give the chemist a real view of how that molecule is interacting with the protein. And the computational chemist can render different types of surfaces, so they can really see what types of interactions the compound has with the target. And from there, based on these interactions, then they can build the types of libraries they need that can have a more rational approach to getting structure activity relationships. So they can really see what modifying, this little ring on here, what it's going to do to increase or decrease the compound's ability to interact with that target or that complex. So I've given you a flavor for what a lot of different roles and expertise that come into like just one lead discovery team. And we really work as a team when we're on these projects. It takes a lot of different people and a lot of different skills and know-how. And that's part of why I love my job is I have all these experts who are so talented to learn from. And it's just increased my amazement with science and with technology. I'm going to skip that. So where are we going next? So we've been doing the high throughput, we've been doing the medicinal chemistry, we've been doing plates and single well things, but that really hasn't bought us the new antibiotics that we really need. So what's going to happen next? And these are just my thoughts, they're not my company's thoughts, so take this with a grain of salt. I'm not speaking for them, but for me, it's that real integrated systems biology. We have to understand how microorganisms interact with their host. What are they really doing when they're in their environment? They're changing their gene expression. We know they can, we know they do. But can we really understand how and how to utilize that? And again, this comes into a harder question because every bacteria in the host is going to behave differently. So is there a common thread that we can use, or are we looking at single agent drugs? And the only way we can really enable that is to have better diagnostics. And there's all kinds of apps that are coming out that measure all aspects of your health that they're just dying to do. When we go to conferences, we hear all about people using Wi-Fi in your home and with those Wi-Fi waves, they can measure your breathing, they can understand your heart rate, they can understand your sleep patterns, they can tell where you're going in your house, how active you are. I can't even fathom all the different things that they can come up with that are going to help physicians better understand you as a patient. And you as a patient can lead to a personalized medicine that's right for you. Because you're complex too. We know that there's extrinsic factors like whether you smoke, whether you're overweight, whether you have other bacterial or other health lifestyle things. Then there's the intrinsic factors, which is just your genetic makeup. And so why when person A goes to the hospital to have a surgical procedure and person B in the same ICU ward with the same sets of nurses and doctors in the same wards, why does A get the infection and B doesn't? Maybe there's something about their genetic and their epigenetic systems that make them more vulnerable. They have other underlying health conditions that make them more immunocompromised. And people are saying it also has to do with their own symbiotics that they have in their gut. There's a lot of things. We are what we eat. There's different studies that are trying to look at how that symbiosis that we have with bacteria in our guts are affecting our overall health. And that maybe that's a different way of looking at antibacterials, or in maybe a pro-bacterial way that will help us be healthier humans. And then the last thing is, if we still want to look at chemical diversity, we have to really tap into the unexplored areas. So I would swear that every company I work for, we pretty much have the same libraries that we've been screening over and over again against every new target. And it's getting tired. We keep finding the same things. Some people call it trolling for chum. And we need to find out how to make different kinds of fish. And one of the computational chemists that I worked with, he put this little exercise together where he took all the basic building blocks of an organic molecule, and he went through all the math to come up with the permutations, and he came up with there's really 10 to the 60 of chemical structures that could exist, but really only screening 10 to the 6. So there's a whole vast untapped space that we don't even explore yet. from a chemical point of view, but how do you rationally build these new molecules? I mean, you just launch a million chemists to make new molecules, but how do you know which ones are going to be active? How do you employ that resource in a cost-effective way? And again, chemical entities aren't the only thing that we should be exploring. I mean, there's a lot of other modalities that are of interest too, and we have been too heavily reliant on our chemical matter in order to try to solve our problems. So, and again, it's resistance, it's a race against time. Bacteria double every 15 minutes. They have a bunch of mechanisms that they evolve to, and we need to understand the mechanisms of resistance that they have in order to better understand how to counteract those as well so that we can stay effective. So my parting thoughts were, when we were asked what is necessary for this career, for me it's to think critically, to question dogma. I mean, when I look at over the years that I've spent in this career, there's lots of things that we said, oh, it has to be this way, you've got to do it this way. And we proved to ourselves over and over again that that's not the only way. We didn't know this, we didn't understand this part of it. And so teaching your kids to ask why, that can be frustrating sometimes, like a 4-year-old where everything they say is why, but to understand too how things work. I mean, there's lots of things that are very mechanical in cells. There's actual physics in terms of what they do and how they do it, that we're not used to thinking of them in those processes in that way. And I really applaud the scientists that are exploring that world, because I think it has a real bearing on how we understand how a bacteria or any living thing survives. And the big thing is forming a testable hypothesis. Can you ask a question? Can you design an experiment that can give you a clear interpretable outcome? Do you know how to set up the controls for that experiment and then observe the data from it so that you can come to an understanding, which may just be, now I have another question. And the last thing is to really work as a team, because in every setting I have at work, I am working with other individuals. We're collaborating. We did an exercise in one of my leadership meetings where they gave us a problem to solve, and we were all spread up in different groups. And they told us, you can come up and ask for help anytime you want. And so some people did, and lo and behold, they gave us clues to solve the problem. And then some were like, oh, you can share your clues with whoever you want in the room. And so, different teams would approach us differently. Some people would divide up, and they would go up and they would get a clue, like as many times as they would hand us a clue, they would pass them out to everybody else. There was one team that just said, no, we're going to do this on our own. They were the last team to solve the problem. And none of us got a penalty for asking for help or sharing our information. It got us to the solution faster, and that was all that mattered. It wasn't that we did it all by ourselves and we were so smart and we didn't need anybody else. We were a team and we shared our information. And lastly, I just want to thank all of you. I owe my whole career to my high school biology teacher. He was my inspiration. He was the person who got me fascinated with microbes. And you all have that opportunity. I have 2 kids in high school, one just graduated from high school, and I know what kind of an impact their teachers have on them. When they come home and they tell me how one of their teachers made them think, how they interacted with them, I can just see them light up. I can see the wheels turning. And you have a rare and unique opportunity. Not everybody in their career has that ability to touch those kids in that way. And I thank you for all your efforts, and if you need help, support, whatever I can do, let me know. Thanks.